everybody, and welcome to today's webinar where we're going to be discussing wastewater microbiology basics. Today, we're really going to focus on wastewater life forms such as bacteria, protozoa, metazoa. We're going to be focusing on building good and healthy floc. And we're also going to investigate the connection between wastewater biology that we're going to be discussing as well as sludge age. I'm Michael Frett, a biologist here at Aquafix, working within the lab. And I'm going to be leading you through this webinar today. So thanks for joining, and let's get started. To start off with this presentation, it's going to be important to remember that wastewater treatment is primarily a biological process. And so this process is going to be made up by a multitude of different microorganisms. These microorganisms can be broken down into three distinct groups, including bacteria, protozoa, and metazoa. As you can see on the slide, bacteria are going to make up the vast majority of wastewater biomass, and that's at 95% of total biomass. Where protozoa and metazoa are going to make up less biomass in comparison at 4 and 1% respectively. However, it's really important to understand that these three groups together are going to be the foundation of wastewater microbiology treatment as a whole. So within a wastewater treatment system, when we focus on bacteria, bacteria are going to be vital to wastewater treatment as they are primarily responsible for nutrient removal and thus the reduction of BOD within a wastewater treatment system. In comparison, protozoa are going to be primarily responsible for the consumption of bacteria as well as algae, and through that, the clarification of the effluent. Finally, we're going to see that metazoa are going to be primarily responsible for consuming bacteria and protozoa. So as you can see with those descriptions, a food web hierarchy is really going to be formed between these three distinct groups. And this food web is what makes up the foundation of the wastewater biological ecosystem. However, what's most important for today's presentation and moving forward in the presentation is the fact that all three of these biological groups are going to be indicators for treatment system conditions and overall biological health within a wastewater system. So we can use these three groups to really figure out treatment system conditions as well as the overall health of the wastewater treatment system as a whole. So the makeup, health, and composition of a treatment plant's microbiology is really what's going to be able to indicate environmental problems and treatment conditions or treatment efficiency within a plant. This includes indicating incoming toxicity, nutrient imbalances within a plant, septic conditions, as well as causes of mechanical problems such as foaming and bulking. Here at Aquafix, we really use microbiological analysis as our primary tool for developing wastewater treatment solutions. We believe that understanding the microbiology of a plant is truly fundamental in understanding any issues affecting a plant's ability to treat waste. And so our determinant for the overall health of a plant really comes down to the sludge age of a wastewater treatment system. Sludge age is going to be the length of time sludge is held under aeration in a particular treatment system. The age of sludge can be a determinant for a wide range of microbiological factors. This is such as bacterial growth in population, as you can see that's marked on the y-axis here on this figure. And we're also going to see sludge age determining floc formation and structure, as well as population densities of various protozoa as well as metazoa. Obviously, sludge age is not going to be always a clear determinant to overall health directly. But this diagram is going to be used to explore the microbiology further, as well as make connections between the microbiology, sludge age, and the overall health of a wastewater treatment plant. So this is going to work as almost our visual aid moving forward in this presentation. In regard to microbiological analysis, Aquafix is able to provide laboratory testing for wastewater samples. Our microanalysis and filament origins testing is primarily focused on filament ID and quantification, explanation of filament presence and its origin, so really what's causing these filaments to grow, analysis of floc structure, oxygen penetration, and EPS levels, which we'll discuss further later, as well as an analysis of protozoa and metazoa within the system, And this all kind of leads us to an overall sludge age and overall health for a particular wastewater treatment system. And so really, yeah, through all these observations, we are really going to be able to conduct a comprehensive analysis of the wastewater microbiological health within a sample and a system, and through that make educated treatment recommendations for specific wastewater systems that we're looking at and analyzing. And so we're going to explore this analysis really further throughout this the rest of this presentation and explain how we conduct our analysis and what we look at when we're looking at a wastewater sample to determine overall health, floc formation, as well as treatment efficiency. And so starting off, our initial observations are always going to begin with bacteria within a wastewater system. As we stated earlier, bacteria are going to be responsible for nutrient removal, and they're going to make up the majority of biomass within an activated sludge system. We're going to have three general categories of bacteria that we'll be talking about. That will be free bacteria, flocculated bacteria, and filamentous bacteria. And when we're talking about bacteria, bacterial health and viability can really depend on many factors. However, it seems that environmental factors appear to be the primary determinant for a wastewater bacteria's ability to reduce nitrogen, carbon, and phosphorus, and thus reducing BOD. And these primary environmental factors are going to include dissolved oxygen levels, temperature, pH, toxicity, and availability of micronutrients. And so these environmental factors are not going to, they're not only determining really the overall health of a bacterial population, but they also can determine the proportional populations of different types of bacteria within a specific wastewater treatment system. And we're going to analyze that a little further later as well. After talking about bacteria in general, we're going to take a look at flocculated bacteria and specifically the process of floc formation. So here in this first image, we can see that we have free bacteria that can be seen dispersed throughout a microscopic slide. Free bacteria can be defined as a single-celled bacteria dispersed throughout the bulk liquid, as bacterial cells here are going to be largely isolated and spread out from each other. In order to form flocculated bacteria, floc formation must begin to occur. As you can see here, free bacteria and wastewater are going to grow and develop as they take in nutrients. Under ideal conditions, bacteria begin to produce and secrete flocculating agents, largely EPS, which we'll discuss further. And thus, as more of these flocculating agents are produced under these ideal conditions, bacteria will grow and bacteria will begin to stick together and form these groupings that we call floc. Um, in this last image here, we can see what it really looks like when we have floc formation fully initiated and we have these bacteria grouped together in these separate floc and really clear from the bulk liquid. We really have minimal free bacteria. And so this floc formation is going to provide bacteria with a strong structure and habitat within the bulk liquid. And we're also going to get efficient settling when we have strong floc formation. And so interestingly enough, floc formation can be modeled on our sludge age diagram. And so we're going to see here first at a young sludge age, we're going to see high levels of free bacteria present. And thus we're going to see low levels of settling and a cloudy effluent as a result. Bacteria simply are going to need adequate retention time in order to begin the floc formation process. So as we move up in a longer retention time, a higher sludge age, we're going to see the floc formation process begin to occur as free bacteria are able to reduce nutrients, start to grow, and start to release those flocculating agents as I spoke of earlier. As floc formation continues, the effluent's going to become more clear as bacteria are now able to settle more efficiently. And now, once we're at sort of a healthy and optimal sludge age within a specific wastewater treatment plant, we're going to observe healthy floc formation. We're going to observe really efficient settling and a clear effluent. And we're going to see when you get this correct ratio here, everything is going to be happy in your wastewater plant in regard to floc formation, clear effluent, and efficient settling. And so here we can see just an image of a microscopic example of healthy flocculated bacteria, where you have variable sizes of floc, variable shapes, and you really have a minimal amount of free bacteria dispersed throughout the bulk liquid. So interestingly, sludge age is not only going to be associated with floc formation, But sludge age is also going to be directly associated with the F to M ratio of a wastewater treatment system, and we're going to see all of these terms sort of interact coming up. So F to M is the ratio of incoming food or influent BOD in comparison to the total number of microorganisms within a treatment plant or the MLSS, mixed liquor suspended solids. Online at actually teamaquafix.com, you can all find a handy calculator in order to calculate the F to M value of your own wastewater treatment system. And so um, using your own F to M value, we can really take a look at sludge age being a determinant of not only floc formation, but also more generally looking at F to M levels within a plant in accordance with sludge age. So we're going to take a look back here at our sludge age diagram, and we can now examine the connection between sludge age, floc formation, and F to M ratios, as I said. Starting off, we can see that at a younger sludge age, we're going to have a relatively low population of bacteria as the growth process is only beginning to occur. And with that, we're still going to have low floc formation, as I stated earlier. And so because of this low bacterial population, low amount of floc formation, we're going to see high F to M values corresponding to younger sludge ages, as we're going to have more proportional food coming into a system in comparison to overall bacterial populations. And so as sludge age increases to a relatively healthy range, we're going to see relatively large populations of bacteria as growth has been able to continue and the formation of healthy floc structure has started. With this, at these healthy, more optimal sludge ages, we're going to see healthy and optimal F to M values within your wastewater treatment system. It's important to note that healthy values in regard to F to M are largely dependent on the specific system and are somewhat variable. But in general, finding the right ratio here for your specific plant in regard to sludge age and F to M ratios is going to lead to efficient nutrient reduction and positive overall treatment of your waste. Finally, things are going to start to get interesting as our sludge age is going to increase to be even older here on the screen. We typically now will start to begin to see problems within the treatment system as sludge age begins to get older. And due to the successively long sludge age, these bacterial populations are going to continue to grow and reduce nutrients. However, they're going to subsequently begin to starve because of that, as F to M levels are now going to become low proportionally due to low levels of nutrients left compared to the high excessive levels of bacteria within a wastewater treatment system. And so because of this, floc are now going to begin to disintegrate as bacteria will actually elect to start to consume these flocculent particles that they've secreted in order to form floc. And they'll consume that instead of the limited incoming BOD left within a wastewater plant at this older sludge age. And so with that, you're going to see floc disintegration, and you're going to see these bacteria begin to starve, and you're going to see a cloudy effluent again, and a lot of problems starting to occur as the sludge age begins to continue to excessively age. And so it's important to remember that there's many variables, including influent nutrient levels, amount of wasting, and sludge age, that all are going to determine the F to M values. And what we're looking at here and what we're explaining are just general trends that we commonly are seeing and observing within wastewater samples and specific wastewater plants that we've been working with. And so now that we know a little bit more about the floc formation process and the bacterial growth process, I want to move into some of the more analytical observations that Aquafix looks at when analyzing floc structure within a wastewater sample that we receive. So when analyzing floc characteristics under a microscope, our primary analysis of floc structure and strength is going to be analyzing the presence of extracellular polymeric substances, or EPS, as I stated earlier. This is one of those flocculating agents that help with floc formation, and the primary one in that. We use an India ink stain to actually analyze EPS levels, as higher levels of EPS within floc are going to repel more black India ink and thus produce a brighter floc field underneath the microscope. The difference in EPS can be visualized through these floc on screen. As on the left here, we're going to observe medium levels of EPS where we have a brighter and more defined floc field, compared to on the right where we're going to see low levels of EPS with a less defined floc field and a darker floc field. And so what we're really looking for is we're looking for medium levels of EPS within a particular wastewater plant typically, as medium levels are going to help aid in floc formation, and problems can begin to occur when you either have too low of EPS or too high of EPS. So we're kind of looking for that sweet spot in the medium range. We also actually analyze oxygen penetration into floc underneath the microscope, as the center of floc can often become oxygen deficient and limit nutrient uptake because of that. So under the phase contrast lens, white and tan colored floc are going to indicate healthy oxygen penetration throughout even condensed floc, and this should be sufficient for nutrient removal. And then you can see that on the left here with a healthy oxygen penetration throughout this entire floc. On the right side, we can see that we have lower oxygen penetration, and that's due to the presence of the darker floc field here and the presence of black spots throughout the center of this floc. And so floc that appear darker brown under this phase contrast lens are indicating these deficient levels of oxygen within the floc, and can also indicate issues of septicity within not only the floc structure, but the wastewater system as a whole. So looking at oxygen really can give us a look at the wastewater treatment system as a whole. Finally, continue on with flocculated bacteria. I want to talk briefly about a specific type of flocculated bacteria called Zuglia. Zuglia are a particular type of floc-forming bacteria that are present in almost all wastewater systems. However, Zuglia can actually become dominant under specific environmental conditions, leading to their boom and growth. Typically, we can see Zuglia becoming dominant within a system under elevated soluble BOD conditions. And under high levels of soluble BOD, Zuglia and their floc can actually produce extremely high levels of EPS. And like I said earlier, we don't want EPS to be too low or too high. So with this extremely high production of EPS, we can see caused by Zuglia, we can see conditions start to arise, such as sliming and bulking from that production. And these images here on the right show the finger-like appearance and structure of Zuglial floc. And so I just would like to take a second to summarize Aquafix's analysis of flocculated bacteria within a wastewater sample here. So our overall analysis of floc, it starts and includes with floc size, shape, and structure. We then can look at EPS levels within a floc, as well as oxygen penetration into floc. And then finally, we can also look at Zuglial abundance within a particular wastewater plant. And so you kind of put together all these factors, and all of these factors together help us determine the overall health of a wastewater treatment system, as well as the overall ability for flocculated bacteria to be able to take up nutrients efficiently and reduce BOD within a wastewater system. And so moving on from flocculated bacteria, I would now like to explore filamentous bacteria, and then also share some of Aquafix's analysis of filaments within a wastewater plant. So filamentous bacteria are typically present in every single wastewater treatment plant at various levels. In these two images, we can see filamentous bacteria growing within the bulk liquid here on the left, as well as filamentous bacteria largely growing within and contained within floc, and not so much within the bulk liquid. So when talking about filamentous bacteria, it's important to remember that at manageable levels, filamentous bacteria can actually provide structure and support floc particles staying together. And because of that, some levels of filamentous growth can often be necessary and beneficial for a wastewater treatment plant. However, it's a tough line to plan, because problems can really arise quickly when filamentous bacteria are present at high levels, and this can be due to rapid growth. And what we typically will end up seeing bulking and foaming issues start to begin to occur when this rapid growth happens for filaments. And so this rapid growth is predominantly caused by environmental conditions that are going to favor filamentous bacterial growth, allowing them to dominate a wastewater system and outcompete the good flocculated bacteria that we were talking about earlier. And that's where you can see in this image, when we have this abundance of filaments, we can start seeing that bulking and foaming issues start to occur from filaments. When analyzing filamentous bacteria, we start with the identification and estimation of abundance for each filamentous species present within a sample. So identification is largely going to be done through filament appearance and the use of a gram or Neisser stain underneath a microscope at 100x magnification. So filaments tend to have unique staining properties as well as cellular structure and appearances, allowing people to be able to actually identify specific filaments from each other. For example, here, we can see on the left we have a gram stain, and on the right we can see that we have a Neisser stain image. And on the right we can see that Microthrix parvicella actually stains with Neisser positive granules. As you can see these purple dots throughout this filament on the right here. And that is one of the defining characteristics of Microthrix parvicella. And so these stains really just help us confirm filament identification, and through that be able to look at treatment system conditions because of this filament. And so after that identification and estimation of abundance, we are really able to start diagnosing environmental conditions that are responsible for the overabundance growth of a particular species. And this is because particular species actually are able to grow optimally under very specific environmental conditions. Finally, using that information, we are able to present a solution for problems caused by filamentous bacteria, such as foaming and bulking, by treating the environmental cause as well as treating the filament. And in this image here, actually, we're going to see this white arrow here is now showing a filament that we can identify as type 0041/0675. And this is a filament that is typically caused by low F to M conditions. Although it does not typically cause problems within a wastewater treatment system, it is often an indicator of low F to M conditions. Moving on, we can see the arrow is now shifted to another filament within the image, and this is pointing now to nocardiiforms. And nocardiiforms are a typical foaming and bulking filament, and is also a true branching filament that we can see here. And nocardiiforms are typically found in sludge that is experiencing high levels of fats, oils, and greases, as well as experiencing old sludge age conditions. And so just being able to identify these filaments allows us to know a little bit more about what's going on in the environment of a wastewater treatment plant. And so now, as we're done with filamentous and flocculated bacteria, I want to move on to a little bit of our analysis of metazoa and protozoa within a wastewater treatment system. So moving on to metazoa and protozoa, it's important to remember that these grouping of organisms are going to be much larger in size, much more complex, but really much less abundant, and take up much less overall biomass in comparison to bacteria within a wastewater treatment plant. Also, it's important to remember that these organisms are not directly responsible for reducing BOD, but still do have many indirect and direct effects on wastewater biology and wastewater treatment health as a whole. And so also similar to floc formation and F to M ratios, we can see that the population dynamics of protozoa are really directly driven by and associated with sludge age. As we will see, higher ordered and more complex protozoa and metazoa appearing at older sludge ages. And so now looking at protozoa specifically, we see that protozoa are primarily consuming free-living bacteria as well as bacteria located on the edge of floc. Through this process, protozoa are actually able to help produce a clearer effluent by facilitating strong floc structure through floc grazing, as well as clearing the bulk liquid of free bacteria. So also the presence of protozoans within a wastewater system are also a strong indicator for sludge age, as I said earlier, and overall health. And so I want to start here and start looking at some of the protozoa that are associated with this young sludge age and what they mean. So starting off with the naked amoeba. The naked amoeba is a protozoan, and it feeds on bacteria and clears the fluid around floc. Amoeba use their false feet from protruding their cell membrane and cytoplasm, not only for movement, but also for the consumption of bacteria. And so amoeba are often dominant during high shock loads of BOD, where there are high particulate levels within a wastewater treatment system. Both of these are common with a young sludge age. And interestingly enough, amoeba, when presented under adverse environmental conditions, they're actually able to secrete a gelatinous shell for protection. And so after the secretion of this gelatinous shell, amoebas are now going to be termed and referred to as a testate amoeba. So you can see here this image, it's a naked amoeba that has been able to secrete a gelatinous shell around it and now can be referred to as a testate amoeba. So their testate amoeba are extremely similar to naked amoeba, but interestingly, they're able to withstand adverse environmental conditions because of that protective gelatinous shell and can often then be an indicator of low levels of DO within a wastewater system when you're seeing multiples of these testate amoebas popping up within a sample. We can move on here to flagellates. And flagellates are defined by their presence of a tail-like flagella that they use for locomotion. And flagellates often prefer high levels of soluble nutrients that are commonly present at high F to M conditions and a young sludge age. Interestingly, flagellates are really not able to compete with bacterial growth rates, and thus this is why they're primarily only seen in high F to M conditions and a young sludge age prior to that explosive growth of bacteria and floc formation process really beginning to occur. Now we can move on to swimming ciliates, and swimming ciliates are going to be indicators of really the start of healthy bacterial biomass and growth, as they are free moving around the bulk liquid, and they actually end up consuming free bacteria within the bulk liquid as these bacterial populations continue to grow and start the floc formation process. Swimming ciliates can help keep the effluent clear through this process. However, when you're seeing an excessive number of swimming ciliates within a particular wastewater sample, this actually may be an indicator of excessive levels of free bacteria present and thus a cloudy effluent as a result, because swimming ciliates alone are not going to be able to clear the entire bulk liquid of all free bacteria. So it's an interesting thing is seeing an excessive level of swimming ciliates really can be an indicator of excessive levels of free bacteria within a plant. So now we can transition into discussing other protozoa, but other protozoa that are more indicative of a healthy and optimal sludge age. What we're going to start with first are crawling ciliates. So crawling ciliates are extremely common in healthy activated sludge, and they're responsible for grazing on the outside of flocks and eating some loosely attached bacteria on the outside of flocks. And this really essentially is cleaning up the floc structure and strengthening floc structure. And so because of this, they actually are often termed the cows of wastewater, as they are often observed grazing on the outside of floc. So they're commonly dominant when floc formation and strong floc structure is present. Ammonia levels are low and pH levels are going to be around neutral. And so because of this, crawling ciliates indicate an older and more sustainable and optimal sludge age in comparison to swimming ciliates, as they're going to be primarily consuming flocculated bacteria rather than free bacteria in the bulk liquid. For stalk ciliates here, they are also going to be common in healthy activated sludge systems with an optimal sludge age. Um, they're really also going to be common when we start seeing strong floc structure that has experienced significant BOD reduction. The stalk ciliates primarily clean up the bulk liquid and are dominant when floc formation is abundant, as they actually use their stalks to anchor and attach to floc to provide some more structure. However, as you can see in the image, we're starting to see colonies form of these stalk ciliates. And when you start to see these colonies form into really large groupings, this can start to be a sign that your sludge age is aging slightly, as these stalk ciliates have had enough retention time to be able to grow these expansive colonies within a wastewater treatment plant. Moving on, we can now discuss attached and tube ciliates. Very, very similar to stalk ciliates, and they primarily are feeding on bacteria, algae, and smaller protozoa within the bulk liquid. And they're going to be indicative of a slightly longer sludge age and sufficient BOD removal. And interestingly enough, you can see in the bottom image here, tube ciliates will actually be able to form the surrounding membrane in response to unfavorable environmental conditions such as low levels of dissolved oxygen. So again, if you're seeing an excessive amount of tube ciliates, it might be a sign that there may be some septicity or low dissolved oxygen conditions within a particular treatment system. Moving on from some of the different types of protozoa and what we see and what they really mean to us, we now can start to discuss metazoa. And as I said earlier, metazoa are often more complex in comparison to protozoa, and similar to protozoa, they often are used as indicator organisms for overall health in a wastewater treatment system. And so metazoa, because of their complexity, are often associated with old sludge ages due to their slow growth and development. Because of this, metazoa are actually commonly seen in lagoon treatment systems, as well as complete mixed systems that activate sludge systems that are operating at an older sludge age. So starting off, we can discuss the broad characterization and grouping that is defined as rotifers. Rotifers are really interesting because they come in many different shapes and sizes, but they commonly share a mastax draw structure and some other general characteristics that make them identifiable as a general rotifer. Rotifers are often observed with cilia structures that are used to funnel and filter feed dead and decomposing matter from the bulk liquid. And that's whether that's dead or decomposing matter is algae, bacteria, or protozoa. And rotifers begin to commonly dominate under low F to M conditions that are occurring at these older sludge ages. And that's largely because they need sufficient time to grow and reproduce within a system. And so one interesting note, if you're commonly seeing dead rotifers within a treatment system, this can be a sign of toxicity. And so what I have here is a video of a rotor that's using its cilia to create a whirlpool and really filter feed on the free bacteria and dead organic matter within the bulk liquid, as you can see here, as this rotifer is creating that funneled whirlpool motion and filter feeding there. Just an interesting look at the active feeding process. And nematodes are primarily responsible for consuming bacteria, fungi, and other nematodes as well, actually. And so the presence of nematodes really is just going to indicate an older sludge age, as again, they need time to grow and develop within a wastewater treatment system. You can see the nematode here tangled up within Floc. And moving on from nematodes, we also have tardigrades that we commonly observe, and tardigrades are actually commonly referred to as water bears, and they have distinctive legs and claws when observed underneath the microscope. Really one of the most interesting species to observe underneath the microscope within a wastewater sample. And tardigrades consume protozoa, metazoa, and bacteria, and they're really hardy as they're able to survive periods of oxygen deficit. But similar to rotifers, tardigrades are sensitive to toxic conditions. So they are often observed in really stable but very old sludge age systems. Finally, moving on, we have an image here of a bristleworm. And bristleworms are also commonly present at very old sludge ages. And a bristleworm's growth is going to be stimulated largely by high levels of MLSS, low F to M ratios, and high nitrate levels. And you can see they're easily identifiable by the bristles that line the side of the bristleworm body. We have here an image of a bristleworm where you can see its unique and interesting movement, as well as its distinctive bristle-lined appearance. So you can watch that here as it moves. And so with all of these protozoa and metazoa within a wastewater system, our analysis here at Aquafix really starts with the identification and estimation of abundance of each particular organism that's present within a wastewater sample. Identification of metazoa and protozoa is going to be much easier than filamentous bacteria due to its relative size and easily distinguishable physical appearance and characteristics. It's important to remember, it's really important to remember that observing a single rotifer within a wastewater sample does not immediately mean that sludge age is old. Rather, metazoa and protozoa abundance levels can really be used collectively to come to a diagnosis of sludge age from the collective of observations and proportions of metazoa and protozoa populations within a sample. In summation, we would like to remind everyone about the importance and purpose of microbiological analysis of wastewater samples. So wastewater treatment is first and foremost a biological process. And Aquafix really prioritizes analyzing the microbiology of a wastewater system in order to understand the overall health of a system. And so through this microbiological analysis, Aquafix is able to propose solutions for problems within a wastewater system based directly on observations from a wastewater plant's biology. Our goal here is to support happy and healthy bacteria in order to facilitate efficient nutrient removal and overall water treatment. And so all of our analysis and observations are conducted at the Aquafix laboratories at the University Research Park here in Madison, Wisconsin. So here you can see some examples of our laboratories and where we are actually doing this analysis on wastewater samples. And so within our laboratory, I wanted to share some of the equipment that we are using during our microbiological analysis. Here at Aquafix, we use a Nikon Eclipse E200 microscope and a DSFI-3 camera for observations at 10, 40, and 100x magnification, while using various lenses, such as phase contrast. Additionally, we use Gramianizer kits for filamentous identification, as well as India ink staining kits for EPS analysis. All of our analysis is done on glass slides. During our analysis of a wastewater sample, we first look at the bacteria. And when we're looking at the bacteria, we're primarily analyzing floc size, structure, and shape. We're also going to be taking a look at EPS levels within floc, as well as oxygen penetration within floc. And then finally, we're going to then look at the filaments within a wastewater sample. We're going to look at filament identification, then filament quantification, and then try and figure out a cause for the origin of growth of this particular filament within a wastewater sample. We then move on to analyze protozoa and metazoa composition, where we are again looking at identification and quantification. And so from all these observations and all this information, we really are going to be able to make an educated determination for a sample sludge age, as well as make a determination for recommendations in regard to treatments in order to improve the overall health of a wastewater system. We can take a further look at Aquafix's wastewater services as our microanalysis and filament origin testing process looks like this. First, Aquafix will be able to supply a test kit that includes sterile sample bottles, liners, sample submission forms, and a cooler for return shipping. So upon receiving a sample at our lab team will immediately begin to conduct microanalysis on all samples received as explained earlier. Typically, a report will be produced within 3 to 4 days upon receiving a sample, and we will deliver a report in hard copy form as well as an emailed copy. And then finally, a conference call can be scheduled with technical reps to discuss findings, as well as treatment recommendations from the laboratory. And so now here is a look at a sample report, which includes observations, images, and descriptions here, as we can see, filament identifications. We also can see here a sludge age determination using that sludge age figure we looked at throughout this presentation, as well as a summary of all findings from the laboratory. We also provide a filament table showing filament identification, abundance levels, and cause of filament growth. And finally, we provide some recommendations for treatment changes and to try and improve wastewater treatment of a particular sample. Here's the breakdown for Aquafix's testing services at the moment, from $425 for a base and typical microanalysis and filament origins testing, as we described throughout this presentation, or $1,200 for 4 sample kits to use at your own pace, whether that be for consistent analysis throughout a year or whenever analysis is needed for a specific or particular treatment plant. Finally, we do also offer treatability studies for specific plants in order to conduct specialized testing on an individual plant's mixed liquor. This helps us optimize our recommendations for a particular plant as well as expected treatment results out in the field. And so with all of our testing services and solutions at Aquafix, it's important to remember that Aquafix prioritizes comprehensive microbiological analysis in order to diagnose problems and provide treatment solutions and recommendations for specific treatment plants. And so with that, I would like to wrap up by reminding everyone that our past webinars are all archived through our website, and a video recording of this presentation will be posted soon to our website. And finally, I would just like to say thank you to all of you for joining me today as we explored a little bit about wastewater biology basics, a little bit about protozoa, metazoa, bacteria, and floc formation, and how it all kind of connects to overall health and sludge age. So just thank you for joining me, and I wanted to offer some time here at the end for anyone to submit a question to the question box, and I can try and answer anything that any of you have on your mind now. So thanks for listening, and submit any questions you have now, and I can try and go through and answer those. Here, if I send a mixed liquor sample for analysis, what is the hold time for shipping before the floc and the sample begins to change, like oxygen levels? So we actually recommend to ship samples on ice, and typically it's 2-day shipping that we provide. So when the mixed liquor samples are placed on ice and shipped 2 days, we end up looking at the sample right away then, and with the mixed liquor being on ice, the sample really should not change much at all over those 2 days. It's a different story if samples are shipped warm, but that's why we recommend shipping on ice to preserve the sample before our analysis. So a question here, do all filaments cause foam? Why is it important to know the actual cause of foaming by microanalysis? So all filaments do not cause foam. There are typical foaming filaments such as nocardiiforms or Microthrix parvicella, but it's important that all filaments are not immediately going to be causing foam. However, it's important to know specifically what filament is causing foam if it is a filament-based foam. Because if we can determine that the filament that's causing the foam is nocardiiforms, then we can provide a specific treatment recommendation for nocardiiforms, where we want to be able to figure out what's causing the growth of those nocardiiforms that's causing the foam in order to really take a look at a treatment recommendation for the cause of that growth. And so really trying to fix the cause of the problem instead of just fix the problem, if that makes sense. So that's why it's so important to know the actual cause of foaming by microanalysis in order to provide a full treatment solution to be able to fix that cause rather than just knock back the foam temporarily. So how many times per week should one be doing a microscopic examination? I think that really depends on your treatment plant and how stable and how your treatment plant is performing, but so it really would be variable. But I don't think it's not something that you would need to be doing every single or multiple times a day or anything like that. But I think a couple times a week should be sufficient if everything is running well at your particular treatment plant in regard to a microscopic examination on site. When is foaming a problem versus just part of the system growth cycle? Well, foaming really becomes a problem when it's actually inhibiting, is oftentimes foaming can inhibit dissolved oxygen from reaching from the air into the water source at a treatment site. And it's really going to be a problem when it's affecting your treatment ability on site. So that's where foaming is going to be a problem versus just part of the system growth cycle. Because obviously there's going to be instances of slight foaming that is going to be able to be remediated without a full treatment plan. But when foaming starts to cause a problem for your own personal wastewater treatment plant on hand, that's when we determine a problem and try and get a fix for it. I just wanted to thank everyone for coming out and listening to this webinar today, and I appreciate all of you giving you my time, and I hope you were able to learn a little bit more about wastewater microbiology, and I hope to be working with some of your guys' wastewater samples soon as well. So thank you so much. Please have a great, happy and healthy day. Thanks.